welcome you to this other session of ours. So we've been talking about the light stage, or you can call it the light dependent stage. Others call it the photochemical reaction, others call it the halo reaction. So today we are going to look at the dark reaction, stroke the biosynthetic phase, or the blackmanns reaction, or you can call it the light independent. So you are saying that it is called the dark reaction because does not require light, although can take place in light also. So it occurs in the stroma of the chloroplast. Now the other side, you say that the light dependent or the light stage, that one depends, okay, that one occurs in the thylakoid membrane of the chloroplasts. Now this dark reaction or the blackman's reaction, this one occurs in what we are calling the stroma of the chloroplasts. So the main pathways for the dark reaction, we have one, the Calvin-Benson cycle, we shall look at it, stroke the 3 pathway, the 2 Hertzsprung pathway, stroke the C4 pathway. Now this dark reaction has got 2 main pathways. One is the Calvin-Benson cycle, stroke the 3 carbon pathway. Then 2 is the Hertzsprung pathway or the C4 pathway. So we shall look at this. Let us begin with the main stage of the Calvin-Benson cycle or the 3 carbon cycle. Now we are saying here is where we find the C3 plants or the carbon 3 plants. We are saying that when talk about C3 plants, somebody can say define the term C3 plants. So C3 plants, those are plants whose first stable product of photosynthesis is a 3 carbon organic compound, and that compound is called the glycerate 3-phosphate. We shall look at it. Now from there I want us to look at this. Now from there, yeah, let us begin from here. Let us look here. There we are saying that the 12 PGA in bracket, the 3 carbon. Then from light, we from light stage you have 12 ATP, then 12 ADP, then we form. Then from there still we have also from light, this part we have the 12 NADPH and the 12 NADP, that is the returning light stage. We form what we call the 12 phosphate, or what we are calling the 12 pigalo in bracket the 3 carbon. We say that in the Calvin cycle, that one involves the 3 carbon what? 3 carbon cycle, or we have what we call the C3 plants. Now we are saying that here when it reaches here it divides. Now the 10, okay, let me begin with here. These the 2 carbons from the 12, the 2 carbons form, so okay, the 2 PGL out of the chloroplast, this moves out of the chloroplast and they form what we call the fructose phosphate, which is the 6 carbon. Then from that one it can form what we call glucose. So then from glucose we have sucrose and starch. Then another pathway, now the remaining 10 from the PGL or pigal, these remaining 10, these ones form several steps. One is the 6 ribose monophosphate, which is a 5 carbon. Then this is from light stage, you have 6 ATP and 6 ADP. When they combine with the 6 ribose monophosphate, it forms what we call a 6 ribose biphosphate, which is a 5 carbon. Then from there, these ones are, this one goes okay, this is 6 ribose biphosphate, which are having the 5 carbon atom, this compound. Now it goes into what we are calling carboxylation, where carboxylation it is worked upon by an enzyme, the enzyme we call Autotrophic carboxylase. This is the enzyme which works on this in presence of carbon dioxide to reduce it to a 3-carbon compound with the one we are calling the pigal. So that has been that cycle. So let us continue with our work. Here we are saying that the triophosphate, in the bracket, the glyceraldehyde 3-phosphate, in the bracket, the glyceraldehyde 3-phosphate is the end product of the Calvin cycle. So somebody, this one can come in objective, that which of the following is the end product of Calvin cycle. We are saying that it is the glyceraldehyde 3-phosphate. We are saying Calvin cycle of photosynthesis because all subsequent reactions can also occur in non-photosynthetic organisms like animals and fungi. So they have noted you where to go. You can visit the biological science page 210 and read on that one. So let us proceed. Then let us look at the main. Let us look at the 3 main stages of Calvin cycle. So these 3 main stages of Calvin cycle. We have carboxylation, where we saw Rubip working on the other 6 ribose biphosphate to convert it to the pigalo. We have reduction and the regeneration, the way you are seeing it there. So without wasting time, let us look at the carboxylation. Now, carboxylation catalyzed by the Rubip carboxylase, the 6 molecules of Rubip reacts with the 6 molecules of carbon dioxide and 6 molecules of water, forming 12 molecules of the 3-phosphoglyceric acid PGA. I've been talking about this, the 3-phosphoglyceric acid PGA, which is the first carbohydrate made by C3 plant. Now, when they talk about, they can say which of the following is the first carbohydrate made by C3 plant. It is the 3-phosphoglyceric acid, or the PGA. Then from there, let us look at the second reaction, or the second stage, which is the phosphorylation and the reduction. I hope you have seen it. We saw up there that 12 molecules of PGA reacts with the 12 molecules of ATP, forming 12 molecules of 1,3-diphosphoglyceric acid PGA and 12 molecules of ADP. We looked at that cycle there, the Calvin cycle, which you have been looking at, and I've directed you to BS page 200 something there. Then from there, we have also what we call the 12 molecules of the NADPH formed in a light stage, reduce 12 molecules of 1,3-diphosphoglyceric acid to form 12 molecules each of 3-phosphoglyceraldehyde pigalo. Now pigalo is 3-phosphoglyceraldehyde, or the triophosphate TP. You can call it pigal, or you can call it TP. TP is triophosphate, NADP, and the phosphoric acid. Then out of these 12 molecules of pigal, only 2 molecules undergo isomerization and several reactions to form hexose sugars, sucrose, starch, and the 10 molecules are recycled to produce 6 molecules of ribose monophosphate. I looked at this up there. Then let us look at the the second major step is the regeneration. Here we are saying that the remaining 10 molecules of pigal regenerate 6 molecules of ribose 6-phosphate when 6 molecules of a 5-carbon sugar, that is the ribose monophosphate, react with the 6 molecule of ATP formed during light stage to form 6 molecules of ribose 1,5 biphosphate, in bracket the Rubip, and the 6 molecules of ADP. So without wasting time, allow me look at the metabolism of the glycerate phosphate GP and the glyceraldehyde phosphate TP or PGAL. We are saying a synthesis of carbohydrates. Now we are saying that the GP molecules are converted to form monosaccharides, e.g. glucose, which may combine with the fructose to form sucrose, then transported in the phloem sieve tube, or can be polymerized into starch for storage or cellulose. Then B, synthesis of lipid. Then you have the glycerate phosphate enters the glycolysis pathway and is converted to pyruvate, which can be converted into the acyl group, which combines with the coenzyme A to form acyl coenzyme A. This can be used to form a variety of fatty acids in the cytoplasm and the chloroplasts. Now, B, we are saying that the GP can also be converted to glycol lipids such as the triglycerides. We looked at this when you are looking at chemicals of life, or esters of fatty acids and the glycol, which are important components of the cell membranes. Then you see synthesis of protein, where we have the GP is converted into the acyl coenzyme A and esters into the Krebs cycle. And enters into the Krebs cycle. So some of its intermediates can produce different amino acids by transamination reactions. We looked at this in protein synthesis. So the amino acids are then polymerized into proteins, which are required for growth and development. Synthesis of enzymes and the structural components of the cell. So I call upon you to actually you can comment, or you can put your question in the comment section. So I will be getting that question and I will be clearing it on air. I therefore sign out. So I want to call upon you people that let us prepare for our second topic, second part of nutrition, which is heterotrophic nutrition. As soon as we complete this, time is now or never, and time waits for no man. I sign out.